Metabolism refers to the set of life-sustaining chemical reactions in organisms. These processes allow organisms to grow, reproduce, maintain their structures, and respond to environmental changes. There are two main types of metabolic processes: catabolism and anabolism. Catabolism involves the breakdown of molecules to obtain energy, such as in cellular respiration. Anabolism, on the other hand, is the synthesis of all compounds needed by the cells, like protein synthesis. Key enzymes act as biological catalysts, speeding up these metabolic reactions. Metabolic pathways are series of chemical reactions occurring within a cell, each step controlled by a specific enzyme. The primary energy carrier in cells is ATP, or adenosine triphosphate. Polysaccharides are long chains of monosaccharide units bonded together, forming a type of carbohydrate composed of multiple sugar molecules. There are different types of storage polysaccharides. Starch, for example, is a storage form of glucose in plants and consists of amylose and amylopectin. Glycogen is the storage form of glucose in animals, highly branched and stored in the liver and muscle tissues. Structural polysaccharides include cellulose, a major component of plant cell walls that provides rigidity and strength, and chitin, found in the exoskeleton of arthropods and cell walls of fungi. Polysaccharides serve various functions such as energy storage, structural support, and cell recognition and signaling. Bioenergetics is the study of the transformation of energy in living organisms, focusing on how cells convert energy from one form to another during metabolic processes. A key energy conversion is the transformation of chemical energy from nutrients into ATP. Understanding bioenergetics involves applying the laws of thermodynamics to biological systems. The first law states that energy cannot be created or destroyed, only transformed. The second law indicates that every energy transfer increases the entropy, or disorder, of the system. ATP synthesis occurs through substrate-level phosphorylation, which is the direct transfer of a phosphate group to ADP, and oxidative phosphorylation, which takes place in the mitochondria and involves the electron transport chain and chemiosmosis. Introductory pathways refer to the fundamental metabolic routes that cells use to extract energy from nutrients and synthesize necessary compounds. One key pathway is glycolysis, which occurs in the cytoplasm and involves the breakdown of glucose into pyruvate, yielding ATP and NADH. The outcome of glycolysis is a net gain of 2 ATP and 2 NADH per glucose molecule. Another crucial pathway is the citric acid cycle, also known as the Krebs cycle, which takes place in the mitochondrial matrix. This cycle involves the oxidation of acetyl-CoA to carbon dioxide, generating NADH, F8H2, and ATP. The electron transport chain, located in the inner mitochondrial membrane, transfers electrons from NADH and FADH2 to oxygen, coupled with ATP production. Photosynthesis, occurring in the chloroplasts of plants, converts light energy into chemical energy in the form of glucose. It consists of light-dependent reactions and the Calvin cycle. The integration of cellular respiration involves glycolysis, the citric acid cycle, and the electron transport chain working together to produce ATP from glucose. Additionally, anabolic pathways use intermediates from catabolic pathways to synthesize necessary cellular components. These introductory topics provide a foundation for understanding the complex and interconnected biochemical processes essential for life.